Thanks. So I will talk about the war on facts. That's what you asked me. And here we see just one example where there is disagreement on the quantification of something. And here you see that it's just not always easy to say what the facts are, right? So who is right here? So you say it depends on perspective. So that's one of the things we have here, that we have different perspectives on the same reality. So I work at the Center for Wetenschapstheorie, the Center for the Studies of the Sciences and the Humanities, which is an interfaculty, interdisciplinary research center in the University of Bergen. It was established more than 30 years ago. And what we actually do there is research on the phenomenon of scientific research and trying to understand how we arrive at knowledge by studying all kinds of domains in the different scientific disciplines. We do that from a double competency, where usually our scholars in the center both have a training and experience in the field they are studying, but at the same time we are informed by philosophy of science and theory of knowledge type of perspectives to critically reflect on the way we produce and use knowledge, on the epistemic, normative, and other dimensions in the production and use of knowledge. So that also includes a critical reflection on the relationship between science and society. So nowadays you cannot submit a proposal in Europe without doing something about RRI, which stands for Responsible Research and Innovation. And you may have heard of that, but what is it? There are different definitions. Here's one. And it said it's a process, a transparent and interactive process. And that process interconnects societal actors and innovators and scientists so that they become mutually responsive to one another with view on the ethical acceptability, sustainability, and societal desirability of new technologies, of the process of innovation, of the new domains of knowledge that we explore, and also its marketable products. So this is just one definition. So there's another one saying it's aligning research and innovation to the values and the needs and the expectations of society. But if you look at how innovation goes, it's completely the other way around. It happens in the laboratories where there is no democratic debate, and all of a sudden the technologies hit society long before there has been a societal debate about the desirability of these products and outcomes. So suddenly we have CRISPR-Cas9 genome editing technology, and nobody ever asked the question whether we want to go that way with technology. And now we have the first edited human beings in China, where the genome was edited with this new CRISPR tool. So responsible research and innovation is a process, as we saw in the definitions. It's also a practice where we try to do better and to make the process of science and innovation more democratic and more responsive to societal values and needs. But it's also a practice of the highest integrity and quality that is required, fitness for the use and the functions in society. And it's also a reflective and critical research culture to which we want to contribute also in the training of scholars and PhDs and students. We have to get rid then of all the perverse incentives that we nowadays have in research, where we are judged on our impact factors and Hirsch index and scientific output and all these things that pervert the scientific process. So there's also a need for reform of science. I already wrote a book on that, on Science on the Verge. You can find it on the internet. So here are some questions that we have to ask in responsible innovation. So if we develop some new technology, how will the risks and benefits be distributed? What other unintended consequences and impacts are there? And have we really mapped them and thought about all the possible societal impacts of new technologies? How might these technologies shape the future and change the future? And is that the direction of change that we want as society? What do we know about these new technologies? And what might we never know about it at the time we make decisions on whether or not to develop these technologies? And then more questions related to the process. So how should standards of safety and standards of acceptability be drawn up and applied? And who decides? And how should risks and benefits be defined and measured? And how do we weigh unknown benefits to unknown risks? Because at the moment you develop a new technology, you don't know the benefits and you don't know the risks yet. Who is in control? Who is taking part? Who has a say? Who will take responsibility if things go wrong? And Ulrich Beck, in his book Risk Society, has introduced this concept of organized irresponsibility in the risk society. So we have a system where we have organized irresponsibility. And if a nuclear reactor explodes, no one can be held accountable. If CRISPR goes crazy and is used for bioterrorism or whatever, who was responsible for that these developments were made possible? There is organized irresponsibility there. And how do we know that we are right? So we need to develop ethics along with technology, but we only invest in technology development and not in ethics development. So it's a wrong balance in the funding as well. We need to progress ethics first before we can progress technology if we want to stay in control. So why are researchers doing it? What are the incentives? It's always to save the world. GMOs, the genetically modified organism, were sold as helping to solve the problem of hunger, but it had nothing to do with the drivers of hunger. So it could never solve that problem. So you have to criticize these assumptions that they use to sell the technology and to attract investors. And then are these motivations transparent? And are they really in the public interest? Or is it just a fairy tale that is being sold? So who will benefit really from these technologies? Will CRISPR cure genetic diseases? Think about it. It's not a business model. That's a one-time intervention and you never earn your money back, right? If you solve in the germline a genetic disease, you cannot make money. So the real money-making model is designer babies and this type of dreams of technology, where the rich can design their offspring. So what are they going to gain and what are the alternatives to achieve the same results? So now let's go a bit more into the use of knowledge in informing societal decision-making, in informing policy. So then we come into the war on facts and what is true and what is right, and how do we know that what scientists come up with is actually reliable and trustable information on which we can build policy decisions. So then we see a first problem. At present, we must admit that most published research findings are false. We are in a reproducibility crisis, not only in psychology and the social sciences, but also in the natural sciences. It's really everywhere now that the probability that a research claim is true, it depends on many things. It depends on the statistical power of the test. It depends on how the experiments were set up and all the rest. And then we see that the research finding is less likely to be true when the number of studies conducted in the field are smaller. so there's not much validation. When effect sizes are smaller, if we talk risks, for instance, of endocrine disruptors like bisphenol A that mess up the hormonal system, then the effect size is very small, but you see it on a very large population that is exposed. But then it's really hard to find out what the truth is about these risks. When there is a greater number and lesser pre-selection of tested relationships, so if you have not looked at all the possible hypotheses, where there's more flexibility in design, definitions, outcomes, and etc., and when there are financial interests, and they are everywhere. The whole research is now not independent, but it's funded by earmarked money and all the rest, and by governments and multinationals, etc. So there is a big problem with reliability of published knowledge. And then there is a more fundamental problem in our ability to know and understand complex reality. And this was first introduced by Alvin Weinberg in 1972 in one of these classic papers. And he talked about questions that can be phrased scientifically, but that in practice can never be answered by science. And he gave the example there of the impacts of low-level radiation, exposure to low levels of ionizing radiation, and that may increase the mutation rate. It damages the DNA, so it increases the mutation rate in exposed organisms. So if you take, for instance, test animals like mice, and you do an experiment, then from the higher dose, where the effect size is much bigger, you can infer and extrapolate what might be the expected mutation rate at a low dose. And in this case, Alvin Weinberg was interested in what the biological effects would be of a dose of 150 milligrams. That was the unit they used in that time for radiation dose. And from extrapolation with the linear model, the expectation was that it would increase the mutation rate by 0.5%. And then if you do your statistics, you can calculate how many experiments you need to conclude with 95% certainty that that dose will give a 0.5% change in the natural mutation rate, which is already quite high. So it's a very small effect size. And then the outcome of this simple calculation was that you require 8 billion replicas of the experiment, where you, in a controlled experiment, expose a test animal and then count the number of mutations, and you repeat that 8 billion times. And then you have enough evidence to prove that 150 milligrams will give 0.5% increase in mutation rate. This is simple statistics, power of a statistical test with small effect size. And then he says, even if we go to the 60% confidence level, we still need 200 million replicas of the experiment. And you all understand that science cannot do this. This scale of experiment is not doable. And even if you do it, there is so much uncertainty when the numbers become so high in whether all the experiments were really similar and all the rest, that you run into other practical problems. So we often see that society expects that science will give the answer, but we forget that on a theory of science basis, that is not possible for all questions. There are questions that cannot be answered by science. And then you have to rely on the assumptions on how you can extrapolate from high to low dose, but you can never validate whether that is the correct way to do it. So then there's the work by Funtwitz and Ravitz, and they came with a typology of risk problems, saying that we basically can distinguish between three classes of risk problems. And the most challenging is what I call the post-normal science category of risk problems, where we see extreme system uncertainty and extreme decision stakes in society. So extreme high error costs if we make the wrong decision based on the incomplete and contested and uncertain evidence. So typical characteristics of problems in this domain are that we cannot afford to wait until the science is complete. Then we are certainly too late and we can run into catastrophic outcomes, and we don't want that. Stakes are high, values are in dispute, and there are big conflicts of interest, both in the research and in the practice. And there are irreducible and unquantifiable uncertainties, not only from the trend science type that I discussed before, but many others. You work with models that cannot be validated. You work with scenarios where you don't know whether they are complete. You work with assumptions that can never be validated. You work with extrapolation outside of your domain of past knowledge, so you don't know whether you go into an uncharted world where the behavior can be different from your complex system, etc. So there are all kinds of problems. So uncertainty in scientific knowledge. Let's give an example where experts are not entirely in agreement. And this comes from the protection of a fresh water resource, an aquifer in Denmark near Copenhagen. And under the new Water Quality Act, this groundwater reservoir needed protection. So the government asked for scientific advice. And they asked the 5 best scientists in the country to make an assessment of the vulnerability of the area to nitrate pollution. So they wanted to know, where is the area highly vulnerable to nitrate pollution? Because there we may have to move intensive agriculture out of that area to protect this freshwater resource from pollution. Now here are the answers given by the 5 best experts in the country. And this is a map with grid cells, and the whole area is 175 square kilometer. And the grid cells in the map that are red, they are the most vulnerable areas of that aquifer. So there you need the highest level of protection, which may mean that you have to remove the farmers from that area, relocate them to an area outside that place. Now you are the policymaker, and you have to just make a decision based on the science. So the science has spoken, but unfortunately science does not speak with one voice. And that is not unique for this case, but that is for every environmental risk assessment. If we talk about climate change, or the risks of endocrine disruptors, or the risk of radiation from fallout after nuclear accidents, it's all the same. There will never be unity in the voices that scientists come up with. So this is typical for risk assessments. But what do you do as a policymaker? How do you act upon this type of information? What would you do? And how do you use this in the court, which may, from a law perspective, be even more challenging, right? In politics, you can maybe negotiate some consensus and work from that, but in the court, maybe it works differently with standards of proof and all the rest. So let's reflect a bit on what uncertainty actually is. And then in all these controversies on risks, you see that there are different ideas and different understandings by the different actors in the controversy on how they perceive the phenomenon of scientific uncertainty. And then you see this especially with those that work in monodisciplinary natural sciences, where they are used to work under the controlled conditions in the laboratory. There the idea is that uncertainty is just a temporary problem while we are working towards better knowledge and towards perfection with better and more measurements and better science. We are in an intermediate trajectory where the science is imperfect, but in the end, it's possible to produce perfect knowledge. That seems to be implicitly assumed in that model. So the idea is that uncertainty is a temporary problem, and we can reduce uncertainty. And many of these proposals also have in the subtitle, reducing uncertainties about climate change, etc. But then in practice, we see that more and more research is done, but the uncertainties don't go away. So we have now more than half a century of climate research with billions and billions of euros investment. And lead author of IPCC reports, they now themselves say there is a paradox: more knowledge, less certainty. You can look it up. More knowledge, less certainty is a paper by Kevin Trenberth in 2010, where he laments on the problem that the more complex we make the models, the more uncertainty we get, and we have more knowledge, but not less uncertainty, not more certainty. So there is a limit to that thinking when we are in more complex issues. But it fits a science policy interface model of speaking truth to power, which may be problematic for complex problems. So then there is the evidence evaluation view, and that was taken by the Intergovernmental Panel of Climate Change. The idea was, okay, in the 80s, there were many contradicting studies, and then the governments wanted to have a solid base to base their decisions on. So together with the development of an international treaty on climate change, the United Nations Framework Convention on Climate Change, there was this new panel, Intergovernmental Panel on Climate Change, IPCC, which was established in the late 1980s and brought their first report in 1990 based on comparative evaluation of research results and building a scientific consensus with all the expertise and all the published literature and looking at the sort of common patterns in the knowledge and then present it as the best of our knowledge, which is the current consensus in the community. But that leads to a focus on only those aspects where you can reach a consensus on. And if we look at the first IPCC report, the policymaker summary does not speak about all the non-linear risks that are in the climate system, like that ice sheets can collapse if they go over a tipping point, or that the ocean circulation can break down, that we have fresh water input in the North Atlantic for more rain and melting water, and then the whole motor that drives the big ocean circulation will stop, and the warm Gulf Stream, which is part of that big ocean circulation system, will go. And there was a lot of knowledge on all that in the detailed chapters, but there was no consensus on these unknown probability extreme impact risks. So they did not end up in policymaker summary because there was no consensus. But if you talk to the policymakers, I come from the Netherlands, and I can tell you a 5-meter sea level rise scenario with the collapse of West Antarctic ice sheet was relevant knowledge to the Dutch government, because then you have to plan, what do we do? Will we build a 5-meter higher coastal protection for the whole coast of the Netherlands? Or will we talk to Germany, can we relocate to your property, right? So this is relevant, but it is overlooked in consensus type of assessment. So then there is a third view, which is the complex system view, which says, okay, uncertainty is just part of the game. It's intrinsic to complex systems, and it will never go away because we have trans-scientific questions, because we built on assumption scenarios and models that have their own epistemic limitations and problems, limitations to our ability to know and understand complex systems. And mathematics does not solve that. So you can make all kinds of magic numbers with your mathematical models, but they are not better than what you put in terms of assumptions. So they are all conditions on assumptions, and then you have the garbage in, garbage out problem, that the weakest link in your model dictates the quality of the outcome. So then we need another type of approach with knowledge quality assessment, which is one of the things I've been working on over the past 20 years, and trying to help science policy interface organizations in doing a better job in dealing with uncertainty and quality problems. But then if we look in the practice, how in practice the science policy interface deals with these type of uncertainties, we see some problems. So often we see a Bayesian approach, where we just take some kind of average and then update with more knowledge if it becomes available. But then there is no epistemic, no theory of science-based reason to assume that the Bayesian average is better than one of the individual assessments by these consultants or experts. Or maybe why not just see them as possible scenarios that you all have to take into account in your decision-making problem and find solutions that are robust under all possible interpretations of the science. But instead you take a Bayesian average and then design a policy only for that average. But then you may have high error costs if you were wrong. Then we have the IPCC approach, or the consensus approach. You just lock these people up in a room and you don't release them before they have a consensus. So by the end of the day, they will be hungry and want to go home. So they come with a consensus. That's what we do on all these international conferences, right? In the end, there is some result, and it's not perfect, but at least you can move on. We can just do the Donald Trump approach and just base our decisions on emotions, tweets, and what else you have, and no longer on scientific facts. So then we can even argue that Brexit is a good idea and these type of things. We can take a precautionary approach where we try to find a policy that at least protects us against the worst case scenario, so that we try to avoid that we have impacts that we don't want. But then the question is, how can you justify the high costs of precautionary measures if we are not sure that it is really needed? Then there's the academic bureaucrat approach. So you just look at which consensus has the highest Hirsch index or the best citation, the highest number of citation. So that is probably then the best expertise. Or you just take the one that you trust most, or you take the one that best fits your policy agenda and don't even ask the other ones. You just hire the one that you know in advance will give you the answer you need. And nowadays, these consultancies are everywhere, and they produce every scientific answer that you want. And you can do the post-normal approach and try to democratize the science policy interface and jointly try to make sense of what we know, what we don't know yet, the unknowns, the uncertainties, the known unknowns, the unknown unknowns, everything, all the possible scenarios, and then together try to work our way forward within this mess of knowledge and uncertainty and see what is a wise way to make decisions in case we cannot give scientific answers that are 100% reliable. We are in an era where you can buy every scientific answer you need. And here's a report from the Netherlands. It's called Science for Sale. It was by the Netherlands Royal Academy of Scientists, Arts and Sciences, what is it? In 2005, so it's already more than 10 years old, and the situation has worsened since, so it's getting worse. But the research here, the investigations they did under many researchers in the Netherlands, they say we see a decreasing public funding of research. So universities and research institutes become too dependent on external research contracts. And then we see that things go wrong. We see that often the design of research, the collection and interpretation of data are adjusted to provide a favorable outcome for the client. So many scientists in this survey said that they ran into that problem in their own work. And then the publication of research findings is sometimes prevented, so we cannot go public with our research findings if one of the financiers is not happy with that knowledge, if the knowledge is unwelcome. And then we have to keep the unwelcome knowledge secret, or there is possibility for competition with other companies, etc. It's not only by contract research by governments, but also all kinds of other interest groups and industry. And then we know from several studies on the practices of the tobacco industry that there is also this infamous merchants of doubt strategy by the industry, where they actually try to manipulate scientific research to create doubt about the risks of their products, to keep them on the market as long as possible. And this is not only done by tobacco industry, but by virtually all industries that market controversial products, and also by the fossil fuel lobby in the case of climate change. And this book by Naomi Oreskes and her colleague Eric Conway analyzes also how they did it on global warming and the various roles of the Heartland Institute, the Cato Institute in the United States, that published their own non-governmental summary of the IPCC chapters with opposite conclusions. So they used the basic scientific assessment, but wrote a completely different policymaker summary, cherry-picking all the things that said we can wait with stopping fossil fuels, and it's not necessary to act, and it's not certain that climate change has anything to do with carbon dioxide. And actually carbon dioxide is a blessing for the world because it fertilizes the plants and we get more food. So from the tobacco industry, from all the studies on that, we know how they do it. They fund research that supports tobacco, and they have a lot of funding, more than the public funds, so they win. They create all these red herrings, where they investigate as many possible causes of lung cancer as possible, so that if you look up in the literature what causes lung cancer, tobacco will only be one of a million of causes, and then it's no longer standing out as something that you have to do something about. Hide the industry role in that research, so it's often hidden. Publish only pro-tobacco research, suppress inconvenient truths, and attack the credibility of your opponents with very nasty strategies. Attack the messenger. Change the scientific standards, also in the regulatory bodies. Disseminate tobacco research to the lay press and dialogue directly with policymakers via front organizations and lobby organizations around the European Commission, European Parliament, local parliaments, and all the rest. So here's another typology of categories of tactics to abuse, to actually do a war on facts. So if you want to do a war on facts, and there are some unwelcome facts, here's the cookbook, what you should do to undermine the credibility of these facts that you don't like, that hinder you in reaching your policy goals. So appeal to emotion, ridiculize the scientists that come with unwelcome messages, say that they are fear-mongers and all the rest. Personal attacks, miscategorization of the argument, the straw man argument, inappropriate generalization, misuse of facts, misuse of uncertainty, false authority, hidden value judgment, scientific misconduct. You can even create fraudulent studies, get them published in the scientific literature, and there are now companies that actually do that for the industry and make big money with publishing fraudulent papers to show that bisphenol A is safe, to show that nano particles pose no risk, etc. And science policy misconduct, putting all your friends in the advice organizations so that they will come with an industry-friendly outcome. So now I will go to a case of war on facts, and it's the case on insecticides and bees. So you may have heard that the bees have some troubles since the introduction of a new generation of pesticides in the early 1990s, that in short time became the most widely used class of pesticides in the world, and now is causing some problems with insects. So these insecticides are so-called systemic insecticides. And here's the famous book by Rachel Carson. I guess you have heard of this book. It's from 1962, and it's sort of the beginning of environmental science and environmental movement. And it was about DDT. DDT was one of the early pesticides that was used in agriculture and to fight mosquitoes and all the rest. But it had a big problem. It accumulated in the food chain and then ended up in top predators like birds of prey and today still polar bears, etc. So if you look into the fat of polar bears, you still find very high concentrations of DDT, even while it has been banned for a long time. But it's an extremely persistent chemical, so it stays in the environment for a long time. And it had tremendous impacts on birds of prey. It weakened the eggshell, and the eggshell became so weak that it cracked during when the birds were sitting on the eggs. So that meant that the breeding success of the birds collapsed, and there was a sort of collapse following the introduction of DDT, a collapse of the birds of prey. But in her book, she also wrote about systemic insecticides that were then still on the drawing table. That was an emerging technology. So that was a technological development going on in the laboratories with no democratic debate on whether we want that, etc. But she wrote a paragraph on this saying, this is a world where the enchanted forest of the fairy tales has become a poisonous forest, because this is a type of chemical that is taken up by the plant and make the whole plant toxic for insects from the inside. So when an insect chews on a leaf or sucks on the sap, it is doomed and will die. Where a flea bites a dog and will die because the dog's blood has been made poisonous. And I guess some of you have pets and use actually these insecticides to make the blood of your dog poisonous. So this is happening now. And I guess at least a fraction of my audience here today has used these on their pets because it's the most effective way to treat this nasty insects. And then where a bee may carry poisonous nectar back to the hive and produce poisonous honey. 2 years ago, there was a worldwide survey of honey samples all around the world, and in 75% of all the honey samples taken from normal supermarkets, they find residues of at least one of these systemic neonicotinoid pesticides. So more than half a century after Rachel Carson's prophecy, this is the reality, this is the nightmare in which we have ended now. So we are in a world where bees actually produce poisonous honey, poisonous to insect. So what are these insecticides? Here we see DDT, and for DDT we needed 27,000 nanogram to kill a honeybee. That's the median lethal dose for those who have a background in toxicology, so where half of the exposed bees will die if you give them that dose. So if we put that to 1 and we compare it to the neonicotinoids like imidacloprid, the systemic pesticide, then we see this is 7,000 times more toxic. You only need 3,7 nanogram of this toxic to kill a bee. So if I have a sugar cube, and it would be entirely made of imidacloprid, and I distribute it equally over the entire world population, then every person on the planet has enough poison to kill a bee. So that's the amount of 3,7 nanogram. So it's taken up by the roots, it goes into the plant, and then the plant becomes toxic from the inside, so the insect will die. But then we see also, this is corn, and it's coated. So the seeds are coated, and the toxic is in the coating. And the amount of toxic in the coating is enough to make the plant months after sowing, so when they grow, the first months, toxic to insects. So no insect can eat these plants. And there's enough in one seed to do that. But then when this was being sown, the bees died. So here we see beehives, and there are many dead bees. And later they find out that it was a little bit of dust that came free during the sowing. And then if the bees crossed that field while they were sowing the seeds, then they collected the dust on their bodies, and that was enough to kill these bees. But later they also found out that it was in pollen and in nectar of all the treated crops, but also in wild plants that were in the neighborhood, because it builds up in soil and it's persistent. Bees are very important, as you may know. So if we lose them, we have a big problem, because we cannot produce food without bees. Only some bulk food, like the wind-pollinated crops. But 90 major crops, which is 1/3 of the world food production volume, depends on pollinators. And then key nutrients, like vitamins and antioxidants, they come for up to 100% from pollinator-mediated plants. So if we lose them, we have to give every person on the planet a vitamin pill from the chemical industry to replace the nutrients that nature produces for us. And I don't know how to do that. We cannot even feed the world, and give them vitamin pills will be even more challenging, right? And then we see that more than 90% of all the flowering plants on the planet also depend on these little insects that pollinate flowers. So also birds that feed on nuts and berries, this fruit set will not occur without bees, so they lose their food source as well. So then here's a beekeeper, and the beekeeper says, listen to the signs and ban the neonicotinoids. But then we run into the same problem as we saw with the aquifer in Copenhagen, because which signs do you need to listen to, right? Here we see different accounts from different scientists on what the role of these new pesticides is in the bee problems that occurred in the late 1990s in France. And then we see that the scientific studies brought forward by the beekeepers, these scientists say that more than 80% of the losses of bees in that period came from these new pesticides. But then if you ask the responsible authorization board in France that allowed this pesticide on the market, they said, no, it's diseases. It's some crappy mite, that the Varroa destructor mite, and our European bees are not used to this parasite, so that is the big problem. And the Minister of Agriculture just says, well, I want to keep friends with everyone, so let's give equal weight to all the possible causes, and then you see how well it works to find as many reasons for lung cancer as the tobacco industry did. So here we find as many reasons for bee decline as possible, and then this will get smaller and smaller in the public perception. And then the producer, they were almost the same as the authorization board in their judgment. And then there were the academic researchers working at the universities and the CNRS, the independent research organization in France. And they said, no, these neonic pesticides are to blame for this problem. So then we come to the problem that what science can address is not the question that we have as policymakers or as society. We have the questions, are these pesticides safe for bees if we use them on the normal way, the normally prescribed way, the label prescription? If we use them normally, do they pose an unacceptable danger for bees, yes or no? But then you're a researcher and you have to research that. How the hell do you do that? That is not something you can address. That's a practical problem. You can only address a technical problem. So the EFSA, the Food Safety Authority, who first allowed these pesticides in Europe, they have a bee guidance. And this guidance prescribes how the tests should be done. And they have, for instance, a 10-day toxicity test where bees are exposed in the laboratory for 10 days. So that's a technical problem. And then you can say, what is the dose where bees will survive a 10-day exposure? And is this dose higher or lower than what we use in the field? And based on this test, they said, yes, it's safe. The bees survive a dose, and the field dose is much lower. So the pesticide is safe, and we can allow it on the market. So technical problem solved, and they said, okay, yes, it's safe. So they assume practical problem also solved. But then, the 10-day test worked fine for the old generation of pesticides, but not for this new class of pesticides. See what happens if academic researchers do not the prescribed test, but look longer. So here we see bumblebees. we see that a 10-time lower dose in each step, and then we measure how long it takes before the 10 bees in the experiment die. And with 200 parts per million of imidacloprid in the sugar water, there's 100% mortality in a few hours, so it's really toxic. Then with 20 parts per million, it takes 14 days to kill them. Then we go 10 times lower. You would expect the daily dose is 10 times lower, so it probably takes 10 times longer, but it only takes 2 times longer to kill them. So there's an amplification with a factor 5 of the toxicity. So here the total amount of poison you need to kill the bees is lower. If you add all the daily doses, it is 5 times lower than here, and it kills them 2 times faster. And it goes on and on and on with each doubling. And you end at the lifespan of the bees. So if they were exposed to completely clean sugar water, they would also die after 77 days. And then you can see there was also an effect for reproduction, and that was 2.5 parts per billion. So really, really low. And much, much, much lower than what came out of safe level in the 10-day toxicity test. But there was a complete mismatch with this type of chemicals. And then if we go below this dose, what happens? They don't die, but there are sublethal effects. And here we see what are sublethal effects. Here's a bee, and it gets a radar antenna. And that means we can track the individual flight of the bee. And they were released in the field, the dot here. And then we see bees that got a sublethal dose and bees that got clean sugar water without pesticide. So the control bees just were flying back to the hive. They see this ditch, this field ditch, and apparently used that also for orientation to find the hive back. But the other bees that were exposed, they have a very strange flight. And the bee ends up here, and not in the colony, not in the hive. And then the bee has no food left and will die. So bees take a little bit of honey from the hive to fuel the flight, and they have another stomach to collect nectar, but they cannot use that for fueling the flight. So when the fuel is empty, when there's no honey left, the bee dies. So a very, very low dose leads to this problem. So this question, how do we test whether it's safe for bees, requires a completely different type of test. Here you see bee with a radar antenna. That was never done before. Nobody thought that that was an essential safety test for pesticides. And it's still not in the new bee guidance. So pesticides are still not tested in this way. So we have the pitfall of lampposting. We look at the wrong problem when asking the question, is it safe? We look where the light is. We have this test on the shelf, so we use it. But it doesn't give us the answer that warrants that we have no problem with these chemicals. Then we have big problems with the tests that were used in the authorization of pesticides. There was no requirement for statistical power. So many of these neonics were allowed because field tests showed they were safe enough. But these field tests did not have the statistical power to prove that. But there was not a legal criterion that they should have that power. So legally, it was right to put this on the market because they fulfilled the required test by the EFSA. But the test was scientifically completely flawed. It could never prove that they were safe. But that's not a legal criterion, that's a scientific one. Then we see big flaws in the experimental setup. The clothianidin, one of the neonics, had an experiment where the hives were 300 meters apart. So they had 2 test fields of rapeseed, 1 hectare each. And then the beehives were put in the fields, and the distance between the fields was 300 meters. And on the basis of this, they said there was no significant difference between the 2 tests, two experiments in mortality, so it was safe. But then if you look at what a honeybee colony does, they forage in a 3-kilometer radius. This is 3 kilometers. So you put the hive here, and this is where they get their food. So if you put one hive here with a small treated field of 1 hectare, and you put the other hive here, and you say, okay, there's no significant difference in mortality, can you really conclude from that experiment that it was safe? No, because we are 100% certain that both colonies were foraging on both fields equally, because they forage in this whole area. So you have to put the other one at least 10 kilometers apart or more before you are sure that they do not visit each other's field. And then they do not only eat from the test field, but in this whole area there are so many other fields where other chemicals are used. So if you put another one 10 kilometers that way, the surrounding is completely different, and the exposure to other chemicals is completely different, and even the food that they collect is completely different. So you can never compare. So there are some fundamental problems with field tests. And then we have conflicts of interest in the regulatory science. And here we saw that the IPBES, the Intergovernmental Panel on Biodiversity and Ecosystem Services, was starting a pollinator assessment in 2015, completed in 2016. One of the authors, it's now this side of the slide, one of the authors, lead authors of the chapter on the impacts of pesticides, had worked for the industry, was someone from Syngenta, and had worked before for Bayer Crop Science, the 2 big producers of neonicotinoids that are suspected to play a role in pollinator decline. So there was a big problem, so we sent a letter to Nature, I was one of the authors, saying that they should have conflict of interest rules for the authors. They said, yes, we were working on this, but they are not completed yet, and we had to start this pollinator assessment, and these were the experts that were nominated by the government, and she was nominated by the UK government, so she ended up as a lead author, and nobody ever checked conflicts of interest. But now they have a conflicts of interest policy, but for this report it was too late. Here's another example, some international committee that advises on the tests that go in the EFSA bee guidance. And we see the effect on bee brood in the hive. 2 representatives of the industry, 3 of governmental agencies, 2 of them went to the revolving door, worked for the industry before, and 1 of a consultancy company that works for the industry. No academic scientists, no beekeepers. And they came up with a threshold saying 30% of loss of the bee brood is acceptable because the hive will have big loss but can recover. And 50% loss of the eggs or other larval stages is an acceptable risk because we tested it and the hive will not collapse. It can recover if we put it in a clean environment without pesticides afterwards. But then if you show this to beekeepers, they say, but then we cannot deliver our pollination services because 1 week we stand here, the next week we stand there, but now the next week we have to send them on holiday to southern France, to a nature area to recover. And then after some months we can use them again for pollination. So this is completely unacceptable. And of course they can recover, but we don't want bees that need to recover. We need bees that can pollinate different crops in agriculture. And this week we are at the apple trees. The next week we are in some other field, strawberries, who knows, etc. So then the war on framing, if time allows. 5 minutes left. So Bayer Crop Science really invested in this frame of honeybee health, and that it's all a problem of this mite. This comes from a brochure from Bayer Crop Science. This is a huge magnification of a tiny parasitic mite, the Varroa destructor mite, that sucks the blood of bees. But then, the honeybee is a managed bee. It's not a wild animal. It's the only bee that is kept by humans. And to say that this is the problem of bees, and it's only this bee, the honeybee, that has a problem with this mite. And we see a decline in all the wild bees. And then we say the laying hen is not a good indicator for the state of farmland birds. You follow this argument, right? If you want to know how is it going with farmland birds, you're not going to the farmers and count the amount of laying hens, the chickens there, right? That is not an indicator for how the farmland birds are doing. How can the honeybee, which is a farmed animal by a beekeeper, right? It's not a wild animal. How can that be a measure for how it's going with bees or pollinators? And from all the bees, it's the only one that lives in a very, very big colony that is really resilient. If you lose half of your bees, it's not a problem for a honeybee colony. But for bumblebees, which only have 250 bees in a hive, in a nest, it's a big problem. But honeybees have 40,000 bees in a colony. It's the only bee that has that problem. So worldwide we have 250,000, no, 25,000 bee species. In the Netherlands, for instance, 350. In Europe, it's about 2,000 different bees. And we see that half of them is at risk of extinction. And then we see it's not only the bees, but all the insects are going down. And there was this alarming study that more than 75% of the insects had disappeared from protected nature areas in Germany over the last 27 years. So we are now in a position that we lose the insects rapidly. And here is the column by Monbiot after that study, saying that Insectageddon, the worldwide collapse of insects, is more catastrophic than a climate breakdown. But then what causes this complete collapse of insects? And then we see there is a war on facts. Here we see the percentage of publications in the scientific literature of 73 papers in the literature saying that that particular cause is the main driver. So we see that 23.9% of the literature says the main driver is intensive agriculture. Then there's 12% says the main driver is pesticides. There's 10% says the main driver is urbanization. Yeah. There is some others say it's fire, it's climate change, it's whatever. You see, there are so many causes, and then the question is, how do we ever know what we should do about it if we don't even agree on if there's a war on what causes it? Then if we look at the story on pesticides, we see that the neonicotinoid pesticides have boomed and have become the most widely used pesticides on the planet. This was 2010, and now we are somewhere here. 40% of the world insecticide market is neonicotinoids. And this is farmland or just land in the US where it is used from 1994 to 2009, and it's still continuing. We always see these graphs saying we apply less and less pesticides to the farmland, but more and more farmland is being treated. And this is only the amount of kilogram. And I showed you the pesticides we use now are 7,000 times more toxic per gram or per kilogram than the old ones. And that is not in this type of graphs. So my colleague Dave Goulson came with this graph in a paper showing the toxicity of farmland to insects and using the honeybee as an indicator. And then we see the trend is like this. So the farmland is being more and more toxic to insects. And the biggest contribution is neonicotinoids. And these are systemic, so these are in the pollen and the nectar of all the flowers on this land. So we did an integrated assessment of what happens if we put these amounts of pesticides in the environment, looking at all the evidence. So we had an update of that in 2017. You can look it up on the internet. And what we found is that it's not only the pollinators that are affected, but many groups of insect species. So we see also aquatic invertebrates and terrestrial invertebrates. So all the animals that live in the water. Let's see, that time is up. So maybe I just stop here. And
hope at the end.
Hope at the end.
Yeah. Oh, thank you so much.
let me see. Yeah,
Okay.
maybe the hope is that we can just do a better job in analyzing these types of wars on facts, these risk controversies, by looking not only at how conflicts of interest here shape what happens in these risk assessment institutions and what happens in how we do uncertainty and complexity in the science, but also how all these things interact. So we need to do critical and argumentative discourse analysis. We have to look at the uncertainties and the quality in the knowledge base, and we have to see what styles of reasoning, how are practical problems translated into technical problems in the institutions, in the boundary organizations that do science for policy. That's my way forward to
Thank you.
shine some light.
Thank you so much. I mean, this has been
read this
incredibly interesting. Thank you so much. We have a break now. If some of you want to sort of stay behind and talk a little bit, I'm sure that Jeroen would allow that.
Yep.
But we'll start the next panel in 10 minutes, so it won't be long. And if you want coffee, you'll have to make the trade-off. But thanks a lot. Det var utrolig bra.
You're welcome.
Og du får jo selvfølgelig en ny kopp. No, it's new. They're very good. So...